I didn't record the last one, but I'll record this one. Okay. So now I'll break you guys into your group. So I'll pull up the Excel sheet so I could break you guys into appropriate rooms. So, okay. Wait, this is the wrong one. It's the other lab. Breakout room 1, 2. So this one's 2. Okay. So I service Jessica in a room. See breakout rooms. So recreate breakout rooms manually.
Hey, Carlos.
Yeah.
I never heard my name called.
I think you weren't here when I called you. So yeah, but I saw you when you showed up.
Oh, okay.
Yeah. Let's see. Okay. Trying to figure out how I'm gonna do this quickly. So because not everyone's showing up. So recreate all rooms manually, automatically, and then separate you guys from there. So I think it is probably due to not being registered with the email or with Cat Courses directly. I think that might be the issue. I'll talk to Dulai to send you guys a message on how. So the registration process for Zoom is uniform. So there's no like difference in how you guys register. So then you guys, so because all the other labs are gonna have the same issue. They're gonna have you guys just like not being able to break out into the appropriate breakout rooms. So let's see. So I have you guys in random rooms. So Anna and Jessica in one room. Jennifer. Move to room 3. Yeah, this is gonna be slow. So while I do this, I'll probably have you guys watch the video. I mean, we're supposed to watch the video after the breakout room, but just so I don't have you guys just waiting, I'll put the video up right now. So let's see. I'll play the video. That gives me 5 minutes to. So it's that video, lab 2, day 2, right here. See if you guys could hear sound. Share computer sound. Can you guys hear?
Yes.
All right. Okay. So I'll let this play while I put you guys in breakout rooms.
Hi, it's Mr. Anderson, and welcome to AP Biology Lab 6 walkthrough. This is on molecular biology. So it's the study of this material, deoxyribonucleic acid, that genetic material that makes life possible. In this lab, there are actually two parts to it. There's the transformation part, where we can transform a bacteria, make it different using a plasmid, and then gel electrophoresis, how we can separate DNA, how we can digest it, break it into fragments, and then separate it according to the length of those fragments. And so let's get started. The two things we'll do will be bacterial transformation. Transformation is when you're taking one bacteria, so let's draw an E. coli that looks like this, and here's another E. coli, and we're transforming this one by passing DNA between the two. In this case, we'll pass a little bit of DNA called a plasmid from one to another. The plasmid we'll be using is something called P-Glow, so it's a way to actually get this bacteria to glow, fluorescent glow under a black light. Second thing we'll do is gel electrophoresis. Gel electrophoresis is separating DNA according to its fragment length. So we're basically running DNA through a gel. It's almost like Jell-O, and then we're separating according to the length of those fragments. So those are the two things that we're doing. The bacterial transformation we're doing is using a plasmid. This is the plasmid right here. It's called P-Glow. Basically, P-Glow is a section of DNA. So in a bacteria, they're going to have their regular DNA, but they're also going to have these little bits called plasmids. Plasmids give them ability to do something. So a fertility plasmid would be an example, or resistant plasmid. So that's going to be one that gives bacteria antibiotic resistance. And so in this plasmid, it's been engineered. In other words, it's been built by humans. On one bit of the plasmid, we're going to have the origin of replication. That's where it's copied. These ones are also going to have ampicillin resistance. So when we grow them on ampicillin, only the ones that have picked up the plasmid are going to grow. Here we've got the glowing fluorescent protein. This is extracted from a jellyfish. And then they also have a trigger, arabinose sugar. So if you add arabinose sugar, they're going to make that glowing fluorescent protein. If there's no arabinose, then it's not going to show up. And so basically what we'll do is we'll have 4 different types of bacteria. The bacteria we're using is E. coli, Escherichia coli. So basically we're going to take some of the E. coli, and we're not going to give it the plasmid, so it gets no DNA. We'll plate some of that on what's called Loria broth. That's just basically food for bacteria. And then we'll plate some of it on Loria broth with ampicillin. So since it has an antibiotic, it's not going to be able to grow. We'll then add the plasmid to some of the bacteria. We'll grow some of them on Loria broth and ampicillin. Some will be able to grow, those that pick up the plasmid. And then we're also going to have some of those with this trigger, this arabinose sugar as well. And I'll show you what all those things are going to look like in just a second, but first let's discuss it. So basically what you get is a plasmid. A plasmid is a little bit of DNA. We're going to add it to bacteria, and so the protocol for doing that basically entails putting them on ice, the bacteria. You then add a transforming liquid. It's calcium chloride is what it is, and that causes them to kind of loosen up their membrane and more likely take in that plasmid. We're also going to heat shock them, so they'll be on ice, then we'll heat shock them, and then some of them will pick it up. And when I do this in class, it's 2 or 3 groups out of 20 or 30 groups will actually get it to transform. And so there's a lot of chance to that. But if you get one bacteria to pick it up, you can get a colony of that, and then it's going to grow. And so basically, let's kind of talk through it. If bacteria doesn't pick up the DNA, what should it look like here? Well, we should have a lawn of bacteria. If ampicillin, we should have no growth here, because unless they get that ampicillin resistance, they're not going to be able to grow. LB ampicillin, the ones who get the plasmid, should be able to grow if they're transformed, and then these are going to be the only ones that actually glow. And so if we plate them on it, what we find is that the ones that actually didn't get the plasmid, but they're on Loria broth, what you'll get is they'll cover the whole of the plate. You can kind of see that on here. The ones who we try to grow on ampicillin, the ampicillin is going to kill them. In other words, it's going to kill all of them, so since none of them picked it up. Likewise, on the ones who have the LB and the ampicillin, the ones that are transformed, we're going to see some colonies. So each of these colonies represent one bacteria that picked up the plasmid. It's able to make copies of itself, and it's able to grow. So you can see not a ton of them were able to do that. But then finally, we get the really cool plate where we've got LB ampicillin, so all these bacteria were transformed, but we also have that arabinose sugar, so they're able to use that to produce this glowing protein, and so you get this fluorescence in the bacteria. And so what do you need to understand? What a plasmid is, how it transforms bacteria, and then how that might be manifested in all the different growth on all the different plates. Second part of the lab is called gel electrophoresis. Gel electrophoresis is going to use a gel box that looks like this. There'll actually be liquid in it. And then you'll have a little fragment of jello. This is an agarose gel. So this is what it would look like. You're going to load up the DNA in these wells on one side, and then they're going to migrate across. They're going to be pulled, since they have a negative charge, they're going to be pulled towards the positive end. And so we let it run for a certain amount of time. Now, if we were to let just DNA run, what you'd get is one fragment being pulled across. The smaller fragments of DNA are going to be able to make it farther, and so the smaller it is, the farther it's going to migrate. So we know that this one right here is a smaller fragment of DNA than this one is. But we also want to trim the DNA, and so we use something called a restriction enzyme. Restriction enzymes come from bacteria. Basically, bacteria are constantly being infected by viruses, so they use restriction enzymes to cut up all their DNA, remove the viral DNA, and then glue it back together again. And so basically, a restriction enzyme is going to cut between the nucleotides. So it's going to cut the DNA in specific points, but it's only going to cut the DNA when it has this specific reading frame right here, where it's GAATTC, and you can look that reads it in the opposite direction on the other side. So basically, we can digest it using a restriction enzyme that's going to cut the DNA up into a little bit. If we were to use a different restriction enzyme, it would cut it up into smaller bits or different bits. And so if we were to run all of this through a gel, well, this would be one big fragment, so this would not move very far through the gel, but this fragment way up here is very small, and so it would be migrating even farther. And then these ones would be, I don't know, somewhere in the middle.
I think I let the video run a minute too long. So we were only supposed to watch it up to 5 minutes, but I think I got all the rooms figured out. So I'll stop this. Okay. So there are 20 people. Let's see. I'll separate you guys into rooms. Let's see. Open all rooms. So assign to 4. Okay, make sure you guys join your groups. So Saul and Tanzil, make sure you guys join the groups. So that way you guys could have discussion within your groups. All right, so everyone's in a group, okay. So message to all, broadcast. So image to Canvas.
ARA. It's the trigger for the P-Glow.
Okay. Yeah. I'm gonna be going through all the rooms. So I think you guys should be able to broadcast using your own computer if you guys want, or you guys could just discuss it.
Well, we're just discussing it.
All right. So I'll just be going through all the rooms. But yeah, just like explain, what do you see? What does it mean? And what does it mean? Like, what is it expressing in each plate? So I'll be going through each room.
Okay. Telling.
Okay. So you guys are all in the group, right?
Yep.
Got the group right?
Mm-hmm.
Okay.
Yes.
So yeah, just explain so if you guys you guys have access to the picture I uploaded, right?
Yeah.
All right. So just open it, discuss what you guys see, what does it mean, and try to answer some of the questions in the booklet. And I'm gonna be going through all the rooms right now, but I'll come back. If you guys have any questions, just feel free to let me know in the group chat or yeah.
Okay.
No problem. Oh yeah. So for Emily, since your lab partner isn't here, and I think the other girl dropped the class because she's not on my roll sheet. So I'll have you in this group for now. And I'll probably have the other lab partner join you guys once he gets back. Because that way we have a group of 4. So for
Okay.
here, you guys could
And then,
wait, go ahead.
I don't know what's happening like with my screen, but it's just black and I can't see anything. So I was gonna call IT about it, but if I call IT, then I have to like leave this thing.
Oh, for right now, it's fine. For right now, it's fine. We're just discussing. So as long as you should, you could just, right now it's fine. You could
Okay.
just discuss, and on Canvas in section 2 for your guys' class folder where I uploaded the pictures, you should find the sample data for this lab, for lab 7. So
Is it under like document 6?
yeah, that one, the PDF.
Okay.
Yeah, that
Okay,
one. You guys
cool.
could just discuss that one for now. So you're fine
Okay.
for now.
So a quick question. From here
Yeah.
to the end of the semester, we're gonna be a group?
Yeah.
Okay.
Yeah, because I think so they had 3 people in their group, but that one of the girls, I think she dropped the class. I'm not sure because she's not on my roll sheet anymore. So yeah, so I think
So it was like before it was me, Max, and then another girl, and her partner had dropped. So
Yeah.
it was me, Max, and the other girl.
now that other girl, I think she dropped the class because she's not
Ariana?
Ariana, yeah.
Oh. Okay.
So yeah, so it's just you and the other lab partner. So yeah.
Okay.
So then once he comes back or he joins back, then he'll just be added to this group.
Okay.
Yeah. So this is going to be the format from now on, and yeah. So you guys could just discuss that image, what you guys see, and try to answer some of the questions on the booklet.
Okay.
And then I'll be going through all the rooms right now.
Okay.
All right. This is room 3.
Question.
Wait, go ahead.
No, nothing.
Oh, okay.
Saying to discuss
He
about
said he
some
uploaded
image
an image
on Canvas. Oh my god, where?
Okay.
Can you guys even hear me?
Yeah, I could hear you.
Oh, okay. Carlos, sorry, but where you upload the image, can we see on the screen, or we have to go to Canvas?
You guys might have to go to Canvas on your personal computer.
Okay, so I should go files and where?
So I'll show it again so you guys can see. Section 2, files in section 2.
Files, and let me see.
And it should be a document labeled.
Section 2.
Yeah. And
Document label what? Doc 6 or 1 GPG or 2 GPG?
Doc 6.
Doc 6. Okay, thank you.
Yeah. And that's your data. You guys could discuss it and try to answer some of the questions you guys have on your I guess the lab 7 questions. So do you guys have any well, I'll just let you guys discuss it.
Yeah. It's on cat courses.
Were you guys able to find it?
I opened it separately on my computer. I got it.
All right.
So
Can you share your screen with us?
You want to share your screen? There's like a green button when you move the cursor around. And I'm pretty sure you guys should be allowed to share your individual screens if you want.
I'm having really bad Wi-Fi.
So the last one has it's like fluorescence, so that means it has that sequence.
A-R-A. And
Is he gone?
yeah, I think so.
Oh, shoot.
I
That's
wanted
what
to
I was
tell
like.
him. Oh shoot! I needed to tell him I'm having really bad Wi-Fi problems.
Oh
it's
no,
fine.
if you have bad Wi-Fi problems, you could call in. I think there's a phone number just in case. So that way you could still listen in.
Yeah, I'm having to use my phone. My laptop like can't work. Like it's not
Alright.
working without Wi-Fi.
All right, so I'm
It's kind of
recording.
hard to do.
I'm recording the whole thing, so I'll be uploading the video too. So I'll give you guys an announcement about that.
Oh, that's
Yeah, because it's kind of hard to keep up. It's kind of
Wait.
hard to keep up with my phone.
All
So
right.
our break room is also recorded too?
Probably.
Oh, Dina, I told you.
Okay, so I'm gonna be going through the rooms, but just discuss it with you guys because I have to go through all the rooms real quick.
But okay, so our main focus right now is just like going over the
The
document presented, right?
Yeah. So like what the video you guys saw and how you guys could apply like how they used to analyze the expression of the GFP and the plasmids.
Yeah. Okay.
All right. So I'll be going. So this is room 4. Gotta go to room 5 or room 6. All right. I'll let you guys discuss it.
Alrighty.
All right. So I got you guys in the right group, right?
Yes.
All right. So you guys, so were you guys able to get access to the Doc 6 picture that I uploaded?
Yeah.
Alright. So just you guys could quickly discuss it and try to figure out if you guys can answer some of the questions, and then in a bit I'll bring it back to the whole class so then we could all discuss it and I could go through it. But just try to see if you guys have any questions. So based on what you guys saw in the video, see if you guys could sort of like explain why certain things are being expressed in the 4 plates.
So on the first plate, it has like a lawn growth, right?
Yeah.
Because it has the broth and it can grow, but it doesn't have the plasmid, so it's not dying. So it's just growing wherever. Whereas in the second one, it's empty because the ampicillin's killing them all because it doesn't have the resistance from the plasmid.
Yes.
The third one, it does have resistance and it also has broth. So just a little bit's growing compared to the first one. And then the last one, a lot of it is growing because there's arabinose, meaning there's extra nutrients for it to grow. And because it has a plasmid, it's expressing glowing.
Yeah, that's perfect. Yeah. That's pretty much it. That's the main explanation for all the plates. And then there you have colony growth instead of lawn growth for that reason. Yeah. Yeah, you guys got it handled. Okay, so I'll go to the rest of the groups. Will you guys discuss it? If you guys have any questions, just let me know.
Okay.
Okay. Did I come to you guys' room already?
Yeah.
Oh, okay. All right. I don't know how long did I give you guys. It says right here, set time limit to 5 minutes. Have you guys been talking for 5 minutes?
Yeah, we were talking for most of the time. We just recently Towards the stopped end, we talking. kind of just everything was said, so we were just
Alright.
kind of sitting there.
Yeah, I'll probably release you guys right now or leave the breakout room. I think everyone's already discussed it. And I went through all of them, so okay. I'll just leave breakout room or how do I stop this? Breakout rooms. Let's see. Close all rooms. Okay, I think we're all coming together again. So I close the breakout rooms for right now. So yeah, so I'll show you guys real quick. I'll just briefly walk through it one more time and then we'll move on. So then I heard you guys pretty much got it down. So where's section 2? Doc 6. So zoom. So pretty much, yeah. So this plate, it has no plasmid, it has no ampicillin, it just has broth. So you just see a blob of growth of E. coli. Here you have no plasmid, but you do have ampicillin. So since without the plasmid, there's no resistance, so all the E. coli dies in this plate. This plate right here, it has E. coli with the plasmid, but go ahead.
Carlos, can you please repeat it again? Because I can, all I have is like a screen on my desktop. So can you just mark like what are you saying, number 1 plate and number 2, so that way I just write down.
Yeah, I could do that.
Okay. Okay.
I think I have this open somewhere. Oh, I could probably draw on OneNote, because OneNote's like, it's like paint. I'll do that. Take snapshot. I'll do new page. Let's see.
Okay, that makes sense.
So I'll start labeling. It's your text right here. Okay, I'll probably just draw it on there. Here. Plate 1, plate 2, plate 3, plate 4. So then how I was saying, this plate has no ampicillin, just the broth and E. coli, so it grows. So no plasmid. This one also has no plasmid, but it does have ampicillin. So without the plasmid, it cannot resist the ampicillin. So the ampicillin or the antibiotic ampicillin will kill the antibiotic, the E. coli. So this is ampicillin is an antibiotic. Here you have some growths, some culture growth. So here you have cultures that have the ampicillin inserted, which is why they're resistant to they have the plasmid inserted, which is why they have resistance to ampicillin, and that's why you see growth in the presence of ampicillin. Here you have plasmid inserted in the presence of arabinose. So you have a lot more growth because arabinose is a sugar, so a little bit extra nutrients on top of the LB broth. And then the ampicillin is not killing it because it has a plasmid inside. And then you also see the expression of GFP protein here a little bit more because there's arabinose that regulates the expression of the GFP, green fluorescent protein. So yeah, so that's for plate 4. So yeah, I think that's the main part. That should help you guys answer the questions.
I have a question.
Go ahead.
Sorry. So LB, the LB is broth, right?
Or like nutrient. It's a specific nutrient broth.
Okay, so why do we need to see the difference between the P-glow expressed and not expressed? Like the difference between LB/ampicillin and LB ampicillin and whatever RA stands for. I keep forgetting the name, sorry.
So between plate 1 and 2?
Oh, yeah. Well, no, between 3 and 4.
Between 3 and 4.
Yeah, I should have said that.
So you want to see how the expression of GFP is regulated with arabinose. So on top of this providing extra nutrients, it also regulates the expression. So this one could express the green, the green protein, but it expresses the ampicillin resistance, but it doesn't express the arabinose. It has the arabinose gene. I mean, not the arabinose, the fluorescent gene, the GFP gene, green fluorescent protein, but it's not expressing it because there's no arabinose. So it's essentially just showing you that this is the switch to turn on the GFP expression. So that's all. That's the difference between 3 and 4. It's just one of them doesn't have arabinose, one of them has it. So you're just turning on the switch. So this one has the switch because there's ampicillin resistance, but the switch is not turned on.
My question is why the first plate has more flattened and then the second one is like pores and stuff going on?
Oh, so plate 1?
Mm-hmm. The plate 1 is like, it seems like the thing is more flattened, and on the third plate we have something.
Oh yeah, so that's like the difference between lawn growth, like in the video, and
on the actual image we're looking at. So number 1 is more flattened, and number 3, where it's LB and AMP, it has more like dots going on.
So
I mean, as if growing.
So that's due to the ampicillin. So
ampicillin. Okay.
So the ampicillin kills bacteria that does not have the plasmid. So when you're inserting the plasmid, not all the cells would have present. If all the cells in this plate, if you're able to insert the plasmid in all of them, you'll see a similar growth to this. But since not all of the bacteria, the success of the plasmid insertion or transformation is very small. So that's why you probably only had like 1 or 2 here, and that expanded. So you have very few successful plasmid insertions. That's why you see spotted. So all the other ones got killed, but the successful insertions survived, and they made these little circles dots. Here, there's no ampicillin, so this is just growing normally. So this one, the reason you don't see small dots is because none of them were killed. Here, these small dots are due to successful insertions replicating. If you let this grow for like a week or 2, it'll look like this eventually. And then these 2 are the same, except that this one has arabinose and it's a little bit more nutrients. It's a sugar. That's why you see a little bit more growth in plate 4 than 3. But yeah, the essential difference between these 2 is the ampicillin, which kills cells here. But if there was ampicillin here, which is plate 2, it would kill all of them. But what's successful, that's why this is pretty much telling you that the success rate of plasmid insertion is really low.
On the
What was that? So yeah, I mean, does that answer your question?
Yes, I might gonna go back to your recording, because if you're saying it's being recorded, because my connection is getting kind of not really good. So, well, maybe I'll just watch later on and see what's going on.
Yeah. So yeah, it's recorded. So yeah, you're fine. It's
okay.
Yeah, it's recording. All right. So now, go ahead.
Real quick, can you explain the like why slide 2 like doesn't really have anything and then slide 3 does?
So slide 2 doesn't have anything because it has no plasmid insertion. So right here, these two are
Oh, I
negative.
see that on the side. Okay.
So this
My
is
bad.
negative, means no plasmid insertion.
So
And
the
then,
second one is pretty much blank, like it has nothing going on.
yes. So this one, so there's no plasmid, so there's no, so the plasmid has the instructions to resist ampicillin. So this one doesn't have the instructions to resist ampicillin. So ampicillin is just going to kill all the cells or all the E. coli bacteria. Here, it has the instructions because it has the plus sign. That means the plasmid is inside some of the cells.
Oh, okay, that makes sense.
So that's what the negative and plus is for these two. Yeah, that's a plus. So no negative, no plasmid in these two. Positive, that there's plasmid insertions in these. So these have
Oh,
instructions.
so we can say on second, it's kind of like negative control because there is no plasmid, so there is no activity going on. But on third, we have plasmid in it, so it's kind of like doing its activity in there.
Yeah.
Okay, makes sense.
Any more questions? All right. So we'll move on to the next part. So we'll start talking about, so we talked about the long growth, why we see each growth in various plates, and demonstrate expression of gene for, okay. So I pretty much went through that after the video. So now we'll start with lab 8. So this image is uploaded on Canvas. I'm recording the session, so you guys will be able to look at that. And yeah. So that's it for that. Let's see. I'll start by discussing. So mini preparation. Discuss what this means to students. Show this. Okay. So I'll go briefly through the protocol. So essentially what lab 8 is about. So lab 8, I'll pull up the lab protocol. So this one, if we would have done it in person, it'd be really involved. I think this was like a 2-week lab. I'm pretty sure it is. Because it involves 2 parts. But since we're doing everything virtually, I'm not sure about the length of what is expected. Let's see. I'll pull up the files. I'll share again. So I'm just pulling up the files. So I'll start talking about this. So this is part 2 of lab 8, lab 7, and lab 8. Okay. So we kind of talked about, so this is a plasmid, the different parts of the plasmid. So what this is for gluing fluorescent protein, the ampicillin resistance, or here they call it penicillin, which is a different antibiotic, but we used ampicillin. And that's right here, that section. The arabinose, which controls the expression of this, is right there. And now for lab 8, so we have this plasmid inserted into a bacterial culture. Now for lab 8, we're gonna take out this plasmid. So we wanna take it out now. And then we wanna measure, we wanna cut it up and measure the fragments like we did before using gel electrophoresis. So we're gonna go through a process of purifying the plasmid, cutting up the plasmid, so this plasmid right here, and then running it through a gel, and then figuring out where there's the scissors that cut the plasmid are called enzymes. They're a specific type of enzyme. Let's see, they have a specific name for them. They're called, I'm blanking out, but they're called restriction enzymes. They're called restriction enzymes. So we're gonna figure out using the gel where these restriction enzymes or these scissors that cut up plasmids, exactly where they cut. Or based on the fragment sizes and different combinations of restriction enzymes that we will use. So I'll explain that, I'll expand on that a little bit more. So I'll show you guys a video before I do that. But yeah, essentially, yeah, so lab 8, so it's plasmid isolation and restriction mapping. So plasmid isolation involves the purification of the plasmids. So purification of this. So you want to pretty much lyse the bacteria and extract the plasmid. And we also did a lab where we extract the DNA. So we ran it through a bunch of centrifuge and we did some PCR and stuff. But for this lab, they don't use CPR from what I read in the protocol. It's just purification. So I'll go through exactly what they did to purify this. So they talk about the purification. Here they use, so they use different chemicals. Let's see, this explains it a little bit better. So the intro to lab 8 explains it a little bit nicer. It gives you a more coherent explanation of it instead of just looking at bullet points. So here you start off by building the appropriate environment in order to extract the DNA. So when you're inserting the plasmid, you might have some plasmid that didn't go inside the bacteria. So there's going to be some plasmid in the broth and outside of the bacteria. So you don't want that to contaminate. So you add something called RNase, and this enzyme, it breaks down any DNA or RNA or nucleic acid that you might be that might contaminate your data. And then you add EDTA, which binds it takes away anything that helps membrane stability. So yeah, so this binds CA2+ and Mg2+, which is required for membrane stability. So it makes the membrane less stable. It removes any RNA in the surrounding area. And yeah, bro, I hear myself echoing. Let's see. Okay. So yeah, so the RNase and DDTA prepares the environment in order to extract the plasmid. And then to extract it, you're gonna need a detergent, so like soap. So soap messes up it messes up any type of membrane. That's why, like, also to draw a parallel, so like for instance, a coronavirus, it's useful to use soap. It's useful to use the soap. It's useful to use the soap to break away the membrane. Let's see. Okay, so I'm just trying to gather my thoughts again. So going back to the detergent, so we use a detergent to break away the membrane, and in order to extract the plasmid that we want to isolate. So yeah, and then drawing a parallel. So like for instance, coronavirus, it has a specific type of membrane that could be lysed through detergents, so regular detergents. So that's why it's important to wash your hands thoroughly, because it will break apart that membrane that the virus needs to insert its DNA into your body. So it's important you wash your hands. And what was I going to talk about next? So yeah, go further through the protocol of how to isolate the plasmid. So yeah, so we use that. Then we use, I'll just keep using the intro because it's easier to gather my thoughts like that. So then we use this acetate, and where is it at? Somewhere around here. So sodium disulfide, sodium. So yeah, SDS is a detergent that dissolves. So that's like the soap stuff. Detergent is just referring to soap, anything that makes suds, causing cell lysis. Sodium hydroxide denatures plasmid DNA and chromosomal DNA into single strands. Potassium acetate and acetic acid, so vinegar pretty much, forms insoluble precipitate of chromosomal DNA, protein, lipids. Unlike chromosomal DNA, plasmid DNA reanneals completely and remains in solution. So your pellet, in this case, is what you don't want. Your plasmid, it coils up and it remains in the solution. And the rest, like the DNA, cell, proteins, and lipids, that stays in the pellet when you centrifuge. And you only take the supernatant would contain your pellet. I mean, not your pellet, your plasmid. So that's the purpose of using the acetic acid and potassium acetate in order to separate those and allow the plasmid to recoil and remain suspended. And that way you could just take out the supernatant and use that, and you run it through something called an alkaline ion exchange chromatography. Ion exchange chromatography. So what this is, I'll pull up an image. So this is essentially just columns. So this is negatively charged. I'll pull up a more accurate one. Because our DNA has a backbone, a phosphate backbone, which makes its charge overall negative. So that negative charge, you're going to use that negative charge to your advantage in order to separate anything that's not negatively charged, like the DNA or the plasmid DNA, and separate and purify it like more thoroughly. So I'll pull up a better diagram that actually shows like correct charges. Where's that? Pulled it up in the last lab. Should probably copy it. This is a nice diagram. It actually showed like a DNA phosphate binding. I don't see it. I didn't write phosphate. DNA phosphate. Right here. So I'll just copy the image, put it in OneNote. So here you see so you're gonna put your supernatant, and you're gonna run it through a column. So let's see, the columns are, you can see the columns better in the last. So you're gonna be running the supernatant through columns like this. And within that solution, you're gonna have your plasmids, and they're gonna attach to positively charged beads. And this is your ion exchange chromatography. That's what they call it. Yeah, ion exchange chromatography. That's what that is. But in our case, since we're extracting, or we're like trying to filter out the DNA, we want the DNA to attach to the polymer beads. But in our case, the polymer beads are positively charged because the DNA is negatively charged. So here you have a positively charged with these functional groups that would allow the backbone of the DNA phosphate groups to attach and stick properly to the beads. And once it sticks to it, you're allowed to run a few buffers and then an elution buffer, which is just a chemical that would allow once you purify it and have all the DNA stuck to the beads by running like a bunch of buffers and running the fluid, you wash it with buffers and then you run an elution buffer that detaches it, and your final solution should just have purified DNA. So I guess this method just allows you to run it through, and then once you run it through, you run it first with all the stuff with contamination. Then you try to wash out all the contamination because the contaminants aren't gonna attach to the beads. So that's the reason for this. So you let it run through. So eventually, you run it through, and yeah, it's difficult to explain without actually running the experiment. You run it through with the plasmid with contaminants. Then you run it through with buffers in order to get rid of the contaminants. And those buffers are specifically to get rid of specifically charged particles. And eventually you use an elution buffer, which detaches the plasmid stuck to the beads, and it allows you to have them in solution, and you can have them in vials and stuff. But that's essentially the concept behind the purification part. So yeah. And then I will also show you guys a video. Okay, I'll talk about restriction enzymes real quick. So once you're done with the purification process, then you have your plasmid and you're ready to cut up your plasmid. So in order to cut up your plasmid, you're gonna need restriction enzymes. So restriction enzymes are just scissors that's cut in specific places. They cut your DNA in specific places. Some restriction enzymes cut it at multiple spots. Some of them cut it at only one spot. And some of them they make cuts that are sticky or blunt. And I'll show you guys that on the slides. So sticky versus blunt cuts. So your slides. So restriction enzymes, they digest so they cut up your plasmid at different spots. And here you could see, so the allyl restriction enzyme cuts it at this spot. This enzyme, restriction enzyme, cuts it at this spot on your DNA. This enzyme cuts it at this weird spot. It cuts it up like in this way, and this is called the sticky end. This spot is this is not a sticky end, this is a blunt end. That means this one the reason it's called sticky end is because it has more surface area to bind. It's stickier. So more surface area that it could bind to anything that's positively charged. So ideally you want your restriction enzymes to cut in this manner. That way you're able to purify them easier. Because the cut provides more surface area for attachment. So here, yeah, you see some produce sticky ends and some produce blunt ends. And the restriction enzymes that we use for lab 8, they're in, I'll pull up the questions right now. To be more straightforward. So lab A, let's see, questions. So lab 8 uses restriction enzyme EcoR1, EcoRV, and SCAL. So these 3 restriction enzymes are used to cut up the plasmids. So first you take some of your plasmid and you cut it up with just this restriction enzyme, just this one, and then you use combinations. So you cut it with 2 restriction enzymes, this combination of restriction enzymes, this one, and then you finally use all 3 restriction enzymes. And you're gonna get different fragment sizes because they cut up at different spots. So by using the fragment sizes you get from these different combinations, you will be able to determine the relative position of each, over each cut occurs based on the the sizes of the fragments that you get. And in order to determine the size of the fragments that you get from these restriction enzymes, you'll be running your cut up enzymes or fragments through gel electrophoresis, and you'll get your different bands. You have your ladder right here. So for instance, for band 1, it's just the plasmid by itself, will not cut up. This one is cut up with one restriction enzyme. Since the restriction enzyme only cuts at one spot, it's going to be the same size. Same thing with restriction enzyme at lane 3. So just to keep it, I'll open up the other one too, just so I could refer to it again. So lab 8 questions. Just so I can keep going back and forth. So lane 2 is equal R1. So then you go to your, I don't like this up here. So lane 2 is your equal R1. So I go back up here. So this is your lane 2. That's equal R1. So you say like lane 8 is all 3 restriction enzymes. So that's your lane 8. You have 3 fragments because you have all 3 restriction enzymes producing 3 cuts. So I'll go back on here to draw it. So I'll insert, draw, shape, circle, draw color. So you have, so for lane 8, you have 3 restriction enzymes. That means there's 3 scissors cutting at 3 different spots. So essentially you have like let's say one's cutting right here, another one's cutting right here, and another restriction enzyme is cutting right here. You have 1 fragment. So this whole fragment's 1 fragment, 2 fragment, 3 fragments. So that's why you see 3 different fragments for lane 8. And then now so I kind of explained a lot already. It's already like, I'm already an hour and a half in. I feel like I'm talking a lot. So now I'll show you guys a quick video on some of this stuff. So I'll pull up YouTube again. So we saw this one already. So I'll show you guys this one. Still half an hour ahead.
Carlos, are we supposed to hear the video because we can't hear anything?
Yeah, I can't hear it either.
This video is part of the iGEM Academy series, A Beginner's Guide. This tutorial explains.
Can you guys hear it now?
Yes. Yeah.
All right.
How to carry out a mini prep. Mini preps are used to isolate and purify plasmid DNA from bacterial cells. The following mini prep protocol was carried out using the QIAprep Spin mini prep kit provided by QIAGEN. All the buffers required are provided in the kit. In this tutorial, we prepared 2 samples in order to balance the centrifuge. An alternative to this is to prepare 1 sample and to balance the centrifuge with a tube containing an equal volume of water. An overnight culture of bacteria is removed from the incubator. 1 milliliter of this overnight culture is transferred into 2 properly labeled Eppendorf tubes. These tubes are placed into a centrifuge and centrifuged at 11,000 RPM for 2 minutes. The supernatant is carefully removed from each tube. Care is taken not to disturb the pellet. The pellet is then resuspended using buffer P1. Before using the buffer, make sure that RNase A has already been added. RNase will ensure that the cell's RNA is broken down. This will prevent the cells from being associated with the waste. 250 microliters of buffer P1 is added to each tube. This buffer destabilizes the cell membrane and macromolecules. This will allow the DNA to be separated from other cell components. The tubes are vortexed to make sure that the entire pellet has been resuspended. The cells are then lysed with 250 microliters of buffer P2. This buffer contains potassium acetate, which allows the plasmid DNA to remain in solution while the other cellular components are precipitated. The color changes from cloudy to blue. The tubes are inverted gently. The reaction is left to proceed for 5 minutes. The reaction is then neutralized with 350 microliters of buffer N3. This buffer contains guanidinium chloride, which prevents protein folding. The tubes are inverted gently. The samples are transferred to the centrifuge and centrifuged at top speed for 10 minutes. The pellet contains all the cell debris, while the supernatant contains the desired plasmid DNA. 800 microliters of the supernatant is then removed and transferred into a QIAprep 2.0 spin column. The samples are put into a centrifuge and centrifuged for 1 minute. The flow-through is discarded. 500 microliters of buffer PB is then pipetted into the spin column. This step is necessary to remove trace nuclease activity when using endonuclease A positive strains, and is not required for strains such as XL1 blue and DH5 alpha. The columns are centrifuged at top speed for 1 minute. The flow-through is discarded. Next, 750 microliters of buffer PE is added to the column. Before using buffer PE, make sure that the ethanol has been added to it. The ethanol washes out from the column any remaining buffers that were previously used. The columns are once again centrifuged at top speed for 1 minute, and the flow-through discarded. The columns are again centrifuged at top speed for 1 minute. The columns are then transferred into empty Eppendorf tubes. 50 microliters of buffer EB is added to the center of each column. This buffer elutes the plasmid DNA. The columns are left to stand at room temperature for 5 minutes. The tubes are then placed into the centrifuge and centrifuged at top speed for 1 minute. This time, make sure to discard the column and not the flow-through. The flow-through contains the DNA. Make sure to label the tubes properly. The mini prep is complete and the desired DNA plasmid has been purified. DNA can be stored in the freezer at -80 degrees Celsius. Thank you for watching. This video is brought to you by the TCD iGEM Team 2015. To go directly to the next video in the series, A Beginner's Guide, please click on
I'll go back to this. Okay. So that briefly talked about like the purification process. Can you guys hear me? Oh yeah, okay. Have my mic turned on. So yeah, that video talked about like the purification process. I saw in the last part that it talked about like the ion exchange chromatography columns. They're like really tiny columns. But the columns that I showed you guys on the picture, they're pretty big. Yeah, let's see. Chromatography column. They look different. They look different from what they look like here. Those are like the tiny versions of this. But yeah, but essentially the same thing. So to purify that, and then to, let's see, the next part is just left of the video. So that was 5-minute video for an hour and a half in. So we talked about restriction enzymes. And I'll show you guys another video because I think we're supposed to watch this video after I was finished talking about the purification process. And then after talking about restriction enzymes, so after purifying the plasmid through the process you guys just saw in the last video, then after that purification portion, then we actually cut it up with restriction enzymes. So that restriction enzyme cutting, which I kind of briefly explained to you guys, I'll show you guys another video talking about the restriction enzymes a bit more. So those scissors that cut the plasmid in specific positions, there's a video on that too. So I'll pull that up too. So it's, yeah, so have to discuss the restriction enzyme. I mean, I kind of showed you guys, so I'll just show you guys the video right now. Then after that, I'll put you guys into rooms again.
Restriction enzyme digestion is a fast and inexpensive way to gather broad sequence information about plasmid DNA. A restriction digest can give a generalized, bird's eye, or macroscopic view of the entire plasmid. It can also reveal useful information, like the purity and conformation of a plasmid, which is important for downstream applications, like transfection. For example, supercoiled DNA has higher transfection efficiency than nicked DNA. While in theory and in textbooks, restriction digestion is straightforward, interpreting the results of a digest in practice is easier said than done. In this video, we will digest a plasmid of known identity with restriction enzymes, run the products on an agarose gel, and analyze the results. We'll focus on identifying the bands produced under different conditions and use the results to make conclusions about the plasmid. Now, let's start our analysis. The first thing you should do is draw out your expected results. While this step may seem unnecessary, it's good practice and it will make your analysis go a lot faster. In this exercise, we would like to verify the identity of a plasmid we received from Addgene. We'll digest the 7.5 kilobase pair plasmid individually with either AGE1 or XBA1, which are both single cutters, and then with AGE1 and XBA1 together. We'll also digest the plasmid with EchoR1, which has 2 recognition sites in the plasmid. So let's draw out the expected results. Here's our gel. Lane 1 is the size marker, often referred to as the ladder. Lane 2 will be the undigested control. Because this plasmid is circular, the migration pattern of the undigested control diverges a bit from the migration pattern of the ladder, which was made with linear DNA. The uncut plasmid typically adopts a supercoiled conformation. That's because these plasmids were isolated from bacteria, and supercoiled is the native conformation of plasmids in vivo because it's compact. A compact conformation allows the plasmid to migrate through the gel at a faster rate than its linear equivalent. This is because it experiences the same amount of electromotive force, but less resistance due to friction. As a result, it runs a little smaller than the equivalently sized band on the ladder. This plasmid is 7.5 kilobases, so let's draw the supercoiled band right below the 7.5 kilobase pair mark. Another conformation of uncut plasmid DNA is nicked. DNA can be nicked naturally in vivo through the activity of topoisomerases. DNA can also get nicked during purification if the conditions are harsh, so it's common to see nicked DNA in your uncut control. When one strand of the DNA is nicked, supercoiling is released, so the conformation of nicked DNA resembles a floppy hula hoop or a hair tie, making it relatively large. In fact, nicked DNA is so big that as it's migrating through the gel, it experiences greater resistance due to friction than even the linear DNA, which can be more flexible than the nicked. Consequently, nicked DNA runs a little higher than the equivalently sized band on the ladder. Since we're still talking about a 7.5 kilobase pair plasmid, let's draw the nicked DNA a little above the 7.5 kb mark. Now that we've drawn our control, let's move on to our digestions. Lane 3 is the AGE1 digestion, which yields a single product. And because it's linear, its migration is consistent with the marker. So we expect to see a band at the 7.5 kb mark. Lane 4 is the XBAL1 digestion. Just like AGE1, it yields a single linear 7.5 kb band. So we expect to see a band in the same 7.5 kb spot. Lane 5 is the double AGE1 XBAL1 digest. According to our map, we would expect two products, a 7.2 kb band and a 300 base pair band. Since these two fragments appear in a 1-to-1 molar ratio, we know the 7.2 kb band will be 24 times brighter than the 300 base pair band, because it is 24 times as large. This means that if we don't have a bright enough 7.2 kb band, we might not even see the 300 base pair band. Lane 6 is the EchoR1 digest. EchoR1 cuts in two places, so we expect two products, a 5.4 kb band and a 2.1 kb band. Okay, this picture looks complete. Let's run our experiment and see our results. Run your gel long enough to get good separation of the bands. Don't rush it.
Okay, so I think that's it for that video. So that one talked about a bit more about the restriction enzymes. They use a lot more restriction enzymes than we actually use for our experiment. We only use 3 different ones and combinations of those 3 different restriction enzymes. And then we use the size of the plasmid. So I'll go through real quick how exactly we're going to analyze that. I'll walk through the questions. So, okay, I'm still sharing my screen. So the sample data that we'll be using, or the sample image, so the gel image that we would be using for this lab is found in the same folder for your lab 8 folder. So this. Here you see the 7 different lanes, or I mean the 8 different lanes and your ladders. Make sure you label your ladders and label the appropriate size. Make sure you label each appropriate size fragment based on your ladder. And okay, so for the questions. So here, I'll have you guys discuss it a bit. Let's see, what will I have you guys discuss about? So I'll break you guys up into rooms to explain what does it mean? Explain what you see in each plate. Why do you see? What does it mean? That doesn't make any sense because that's exactly what it says for the last portion. Last time I had you guys break up into explain what you see in each plate. There is no plates. It's just explain what you see in the plates from last time. Okay, yeah, no. So I'll just keep explaining this. So for here, you have to make sure you write down what size fragments you observe based on your ladder. So you're gonna label your ladder and then try to correlate the size here with the size of the fragment you have in your actual lane. So like lane 1, you'll have something around here, and then just keep going and make sure you label all of them. And then by knowing the different combinations, so by knowing, let's see, these are the same file. So by knowing that, so these are all single restriction enzymes. So that's why it's only cut up in one place, and that's why they're similar size. Because when you cut up the circle, regardless of what place you cut it up at, if you're just making one cut, you're gonna get the same size fragment. You're just gonna get a straight line, regardless of where you cut. But then you start cutting up in different spots, you'll see that you get different size fragments. And using this, you'll be able to determine the relative position of each cut. And I'll walk you guys through how to determine that. But first, we'll go through the questions, what it's asking. So that's what you'll need to do for this first part. You'll need to determine like the relative positions of where each cut occurs based on the fragment sizes you get from each cut and combination of cuts. And then here you're gonna, I'll break you guys up into rooms and have you guys talk about these questions, and then I'll go through and answer any questions you guys might have. So yeah, I'll do that right now. And then after that, I'll walk you guys through how to determine this portion right here. So yeah, so I'll break you guys off into rooms now. And you guys could talk about the Lab 8 questions here. And then this part, the analysis part, I'll go through it with you guys, but I'll have you guys do the analysis for yourselves, but it'll be recorded on how I perform the analysis. So it'll be pretty straightforward. Okay, so I'll break you guys off into rooms now. Breakout rooms. So open all rooms, and then I'll keep going through and talking to you guys.
The first one, what I knew about is because column is basically, it's like if you see the fragments, the larger the fragment, the more time it will take. And then charge is pretty much going to be same because we have positive beats, and then our DNA or the plasmid is negatively charged. So it will go towards the positive field.
Oh yeah, make sure you guys join Dina and Amy, because because I think you're not in the group yet. So make sure, Tanzil, make sure you join. Oh yeah.
I'll check out. And I checked out for March 29th. I was planning on moving out March 29th, but they emailed me 4 days before my move out day saying that I can't come back, that my card, and I think that number 3 is, I don't know, number 3 is a little bit difficult for me.
Number 3.
Let's try to keep the convo on the lab for right now.
We're just venting our frustrations right now.
Nah, I feel you guys, because like that moving out stuff is it's pretty wild. But yeah, since I'm recording, yeah, that might be inside the recording.
Okay.
So
I have a question for you, Carlos. Like, so I didn't hear you clearly when you responded to the person who asked about Lab 7. So is it a report or just a write-up and answering the questions?
I think it's a report. It's whatever the rubric says. If there's no rubric and it's asking for a report, I'll make sure there's one, because all the other labs have been having like rubrics on what to do with the lab, unless he wants you to follow the same rubric as before. But I'll double check, and I'll send an announcement.
Thank you.
Yeah, just make sure you guys, if you guys have any questions on the lab, make sure you guys figure out. I'll have office hours are going to be at the same times, and they're going to be through Zoom. So I'll also send out an announcement with the link, the Zoom link. But yeah, just make sure you guys discuss the questions right now. I'm gonna be going through all the rooms.
Okay.
All right.
Thank you.
Escape the gel from the other end. So it would just go past the wells and out, probably, depending on how long you left it. So it's not a good idea, because that's when you have to redo it.
You guys are talking about switching the electrodes?
Yeah.
Oh yeah. Yeah, that sounded right.
Oh, I have a question about Lab 7.
Yeah.
Is it a full lab report or just like a...
Yeah, I'll check right now.
Okay.
Because you guys are the second group that asked for that. I'll make sure right now. Because the lab 7 does not say, I'll look at assignments. So lab 7, let's see how I'm supposed to grade it. Check speed grader. Yeah, I think it's a full lab report because I have a discussion, results, intro. Yeah, so I have to grade it based on view rubric. Intro,
Okay.
yeah, it's a full lab report.
Okay.
So there's intro, conclusion, concise manner, style, proper headings, and report organization. So yeah. So you guys have to do a full report for lab 7.
Okay.
So I'll make a full announcement, but yeah.
Thanks.
No problem.
What question were we
on?
You guys have questions?
No. Okay, thank you.
All right. Yeah, I'm just going through all the labs or all the groups. All right. This is 3 and 4.
We're talking about in the plates or whatever. Remember how the fourth one had like green little dots? I think that's what.
That was something else. This chemical is something else. I'm pretty much sure it has something to do with the charge or something. But if it's saying we're gonna visualize it, so that means it has maybe some kind of visualize effect or something. Okay, we can just leave this for now and we can ask Carlos later on. Let me just pull up and see the other question. If one accidentally connected the electrodes to the, well, if you gonna accidentally connect to So pretty much you're not gonna see any migration, right? Because the whole purpose of lab is to make it migrate and get the different 5 fragments. And if you have negative on the side of what you call, I mean, I would just say like it's not gonna show migration of the fragments.
Yeah.
Right?
Mm-hmm.
You guys have any questions?
I think I'm still a little confused about the first one. Like, how does the exchange column work exactly?
Okay, so I'll pull up. So the exchange column. So there's like these charge beads. So an exchange column kind of looks, there's like these small beads, and then these beads will like, so it's like this. So you put in like your sample through the beads, and the beads will have a specific charge, and they're like little magnets. And depending on what kind of what you want it to what you want to stick to it. So like for instance, in our case, we want positively charged beads because the DNA is negative, right?
Yeah.
So, and it depends on how if you oh, okay. I'm like mumbling too much, but since the DNA is negative because of the phosphate backbone, so let me see. Oh, I have it already up here. So in this image over here. So you have those beads in a column, and you have them specifically. You have them customized specifically for what you want to purify. So you want all the DNA and only the DNA to stick to it. So you want it positively charged. So that way, anything that's not negatively charged won't bind to it. So this has a specific charge that will be sticky for this part. If we want to stick to it, because it's they're, I guess it's the opposite. So negative and positive. That way you get a good reaction. And then the ion exchange process. So I guess it's a little vague the way I'm explaining it. So I'll just open this up. So ion exchange, it separates ions and polar molecules based on affinity. So DNA is polar, so it's negatively charged because of its because of the phosphate backbone. The phosphate's negative, and that's what causes it to be polar. And then the way it does it is there's here. So there's an ion exchange that causes it to stick to the beads. So those little beads that you see here.
Mm-hmm.
So there's an ion exchange. Where is it? It talks about it here. On the electrostatic interaction between your negatively charged phosphate groups of DNA, as well as the cationic sites of the chromatographic matrix. So the positive. So there's the interaction between these two, but I want to make sure I have the interactions with the Cl- and Na+. But the interactions with this are pretty straightforward. This positively charged part and this negatively charged part. Because your plasmid is just negatively charged. It's just regular DNA or RNA, depending on what you're inserting. Plasmids are usually RNA because it's circular DNA. Let's see, possesses. No, we don't use HPLC. This is extra stuff. Phosphate groups, the electrostatic interactions, chromatography. I guess it's just to make, so the charge, this charge, the reason the fluid around these two are charged is because it allows, it's like electrolytes. Yeah. Essentially, it's just to have an electrostatic, because these are electrostatic interactions. So you have an electrostatic solution for it to occur. Those stranded and ion exchange chromatography, DNA preservation depend on electrostatic interactions between your negatively charged. Yeah. Doesn't go too much into detail. I didn't read much about the ions in the solution on the protocol, but I'll double check. But yeah, so essentially that's what it is. It's just the interaction. It's a way of purifying the plasmid by allowing it to stick to those columns. So when you're running the plasmid with a bunch of contaminants, you run it through and then you wash it. And when you wash it, all the DNA is going to stick to those beads.
You're washing it with buffer?
Yeah, you wash it with buffer, and then that buffer will get rid of the contaminants. And then whatever's left over, it's just the beads and the DNA. Because it sticks extra hard to the beads. And then to separate that, you add a elution buffer. It's a specific type of buffer that separates the DNA and the beads. But yeah, essentially that's what it is.
In the end, would you have to separate the DNA from the beads?
Yeah, so that's so you'll have at the end you'll have like an elution buffer. So this is just a buffer to clean out like all the stuff. So like let's say for instance this is the DNA that you want.
Yeah.
This stuff you wash it out with buffer, different types of buffers, so it removes it. And then after you isolate your DNA is the only thing that's stuck to the beads. In order to remove it, you'll have to add something called an elution buffer. So you'll just run another buffer through it that will remove the DNA from it. And I think it's like a salt solution, I think is what they talk about. I'm not sure what they use, but they use something to remove, or like I guess that's the reason for the Na plus and the Cl, is they use the salt to separate it, because you'll just have some of like the negatively charged salt attached to the beads and the positively parts of the salt attached to the phosphate group. So that way you'll be able to take out your DNA. But yeah, I'll keep going through all of that. But I'll probably say this again to the whole class right now.
Okay, thank you.
No problem. How do you change? Let me stop share. This is room, you guys room 6? Yeah. I think I went through all the rooms now. Yeah, I think I went through all the rooms.
Okay.
Well, so I guess, yeah, we'll start again with the whole class.
Okay.
Yeah, you guys aren't seeing my screen. Okay, so I let you guys talk for like a few minutes about the lab. I'll go through it again real quick. I'll go through the questions to see. And do you guys have any questions on the lab right now? On lab 8?
How?
So yeah, I'll just briefly skim through the questions one more time. So let's see, I'll share my screen. Okay. So I'll give you guys a little bit more background. So DNA purified with the aid of ion exchange column. So I kind of briefly talked about it. I'm gonna make an announcement with the video also talking about the ion exchange columns. So I'll go through this again because it's kind of confusing. It's not as intuitive unless you're actually performing the experiment hands-on. so that's it sucks that we don't get to perform it, but I'll explain. I'll go through it one more time. So the ion exchange column. So essentially you have a tube with beads. So I'll probably draw it for you guys. Let's draw one. Let's see. So draw shape. I'll draw some rectangles. You have your ion exchange column, and you have a bunch of little spheres inside of it. You have a bunch of these little spheres. And then all these little spheres are positively charged. So they're gonna have a positive charge. And you're gonna run, you're gonna pour your plasmid into the sphere, and there's gonna be an opening at the bottom. This is hard. I flip this upside down. Okay, I don't know. I'll just copy and paste the picture that's right here, so it'll be easier to explain. And then I'll draw on this to explain it. Paste. Okay. So you have these columns with these beads, and you're gonna run your solution with contaminants. So you're gonna run your plasmid, the supernatant you get out from trying to purify your plasmid, you're gonna run it through these beads with buffer. So these beads are essentially made in order to attract and grab the plasmid. So in our case, the beads are positively charged, and they're gonna grab on to the negatively charged backbone, the phosphate backbone of your plasmid. So these phosphate groups are gonna wanna attach to the beads. Then once they attach and all like the contaminants get removed, you're gonna wash it with buffer solutions in order to get rid of anything that's not attached to the columns or to the beads, I mean. And then once you remove those, you're able to then run a few more buffers to fully clean it out, and then finally separate the plasmid from the beads. So to make them, to separate them, you just need to add like a salt solution. So some of the salt will competitively, they'll compete for the spots that are on the beads. So all the negatively charged, like for instance, they use salt, sodium chloride here. These negatively charged salt, chloride, is gonna attach to the columns, and it's gonna compete with the DNA, and then some of the plus, the sodium, is gonna attach to the phosphate group. So that way it'll make it easier to separate the DNA from the columns once you've purified it and cleaned the columns out. And this will allow you to finally clean out the DNA and have a clean solution of DNA or plasmid. So that's essentially the concept behind the ion exchange chromatography.
Could you please explain the Na positive and Cl negative? Because I got the concept like our beads are positively charged, so our DNA will attach because it's a negative. And then we try to remove the contamination, which should be a positive charge. But what's the role of Na positive and Cl negative in there?
So the purpose of the Cl minus and Cl and Na plus is, okay, so at the end, you're gonna have your DNA attached to the beads, right?
Yes.
But in order to separate them, you don't want them to be connected anymore. You're gonna have to give them another option, which is so instead of the negatively charged potassium backbone or phosphate backbone of the DNA attaching to the, it's gonna always attach to the columns, to the positively charged columns. But if you give them another option, like, okay, the DNA could also attach to this positive charge, then it's going to be loose. So that it's not going to be attached to the column, and it's going to make it easier to separate. And then same thing for the Cl minus. It's going to want to attach to the column, and it's going to pretty much remove a spot for the DNA to stick to. So this is just used for this to attach to this, and this to attach to that. So
So what you're saying is attaching Na positive with the phosphate group will help us to separate the DNA from
Yes.
the positive bead, right?
Yes.
Okay, makes sense.
So that's the purpose of something called the elution buffer.
Oh, okay.
So it's to separate the DNA from the filter, which is the beads. It's like the filter.
Thank you.
And then, yeah. So that's for question 1. And then for question 2, which organisms produce restriction enzymes? There might be some info on that on your packet. If not, you guys could look this one up. How is DNA visualization made possible in the presence of ethidium bromide? So ethidium bromide is a fluorescent compound. So it's used in gel electrophoresis in order to make the sample, or like the well or the sample in your well glow. So when you try to take an image of it with UV light, it reflects a lot of light. It enhances the signal. So I could, just a quick Google will give you like a little bit more in-depth information. So yeah, it's just agent commonly used as fluorescent tag in molecular biology laboratories, techniques such as Argos gel electrophoresis. And that's what gives the gels that we run in this lab. So let's see, pull it up here. That's what, oh no, that's the wrong one. That's what makes these gels glow. Okay, so I should probably hurry up so I can have time to explain the analysis. So then if DNA are the same length, so this is just a fluorescent protein that helps for gel electrophoresis. And not a protein, but like it's just a chemical you add to the DNA so it could attach to it and allow you to see the fragments a little bit better. And it reacts with UV light. If DNA molecules of the same length from 2 different sources are run in the same well of the gel, what would you see? So the gels separate based on size a lot of the time, and based on size and based on shape. So on one of the videos you saw that like the plasmid sometimes coils up, and it's not a fragment. So fragments are going to travel in different rates as something that's coiled up and condensed. So shape has a lot to do with how far the plasmid or fragments travel. So fragments and plasmids are 2 different things. So a cut up plasmid is a fragment, even if it's just one cut. And a plasmid, that will coil up and it'll condense. It'll coil up like you saw the girl twisting the rubber band. So that's what the plasmid does. And if one accidentally connected the electrodes to the electrophoresis. Go ahead.
So for plasmid, you're saying when we cut it, it's always gonna coil up, like, because I got the concept. You're saying it's being length-based, and it's pretty much gel electrophoresis is like the shorter the fragment, it's gonna elute first.
Yes.
But what do you mean by coiling in this, especially for this lab?
So like, coiling. So when you have like the circle. So a plasmid is a circular. So without the restriction enzymes, so your lane 1, I'll show you guys right now. So in lane 1, this lane, you have your plasmid, and it looks slightly different from these 3. So these 3 are cut up, they're cut up plasmids. It's only one cut, so it's one long length. So there's slight difference, very hard to tell, but it's not as significant here. You can't really see it too much. But generally, there is a slight difference when it comes to the plasmid, because the plasmid coils up. So let me see if I could pull up an image of a coiled up plasmid. Oh. So yeah. So you have here you see like the plasmid. So you have fragments here, nicked, linearized, and supercoiled. Supercoiled will travel a lot more.
Because it's now shorter in size, right?
Yeah, so it's yeah. So it's like a lot smaller than the linearized. Linearized, you're gonna have a lot of fragments that aren't able to travel a lot. Nicked means it's not gonna supercoil. So it's not gonna like get all coiled up. So if there's like a little piece ripped off. Linearized means it's completely ripped.
Can you go back to our actual observation, like the letters?
Yeah.
So,
You guys see this?
yeah. So what we're saying is on based of that, because we are getting different, this letter 1, letter 2, letter 3, they're on like different size. So because the first 4, they're pretty much linear, they have like same size. So what that means, so they weren't super coiled because they didn't move farther away.
So this one is supposed to be, but since I guess maybe the fragments aren't big enough to see the difference.
Okay.
This one should travel a bit further. It's slightly further, barely noticeable. Maybe if you let the gel run a little bit longer, you might see a bigger difference. Because this looks like they didn't, like the ladder is not really separated too much. So it looks like they didn't let it run for like, they could have let it run for 20 minutes more and you'd see a bigger difference between one and then these 3. Because these 3 each have 1 cut. So these ones should be the same regardless of the amount of time. But this first one is your plasmid without restriction enzymes. So, okay. Essentially, it's supposed to be the supercoiled one. So it's supposed to travel a little bit further. In the image that we have, it's not too noticeable. So this is a supercoiled one. These 3 are just 1 cut. So these would be considered, why did I keep clicking on it? These would be considered the linearized ones. And then the supercoiled would be your lane 1. But since they didn't let it run too long, you can't really see too much of a difference. There's a very subtle difference, but not enough to be able to say it's different. So yeah, this is subtle. Like this looks a little bit ahead.
That's kind of vague because if we are saying it's the number 1 is supercoiled, it should be like way far from all other letters, like 2, 3, 4.
Yeah, that might be due to not letting the plate run for long enough.
Okay.
So maybe the plate wasn't ran long enough. So maybe the difference isn't too much.
And what that means, like if we have 3 different bands, like on 5 we have 2 bands, and then on 8 we have 3 bands. What that means? So that means there was 2 cuts.
Yeah. So 8 uses 3 restriction enzymes. So I'll go to the questions. So lane 8 used 3 cuts. So here you have 3 cuts. For 5, 6, 7, you have 2 cuts. So that's how you see 2 fragments in each. And then for 2, 3, 4, you have 1 cut. So regardless of where you cut it, these 2, 3, 4 should be the same size because it's the same plasmid. It's just a different position where you cut it. But the fragment should be the same size for these 3. These you'll have different sizes. And then in order to, so for the analysis for the fragments, I'll go through that. Is class over like at 5:45, 5:40? So yeah, so I'll go through that real quick before class is over. So I'll open up. Okay, so you guys will have to determine the size of these fragments based on the ladder. So for instance, let's just say, for example, this is 10. So these 4 kind of look like they're at 10, but this is just a made up number. I'm not actually measuring the ladder. I'll let you guys measure the ladder. But for like, this is just like a rough explanation of what the analysis should look like. So I'll draw, what is this? So I'm calling this 10. So this is 10. This is 10. This is 10. This should be a slightly different. It should be a lot further down, but they didn't let it run it long enough. But let's just say these three are 10. Then this one, these fragments should add up to 10. These should add up to 10. These should add up to 10. And these three should add up to 10. So since this is 10, I'm saying, like, for instance, this one will probably be 8. These are just made up numbers. I'm not actually making the ladder. So you guys will actually have to use the real numbers. So if this is 8, then this for sure has to be 2. And then you could confirm it by looking at the ladder, because it should match up these three. So this is 8, this is 2. And then to figure out which one it corresponds to, we look at our lanes. So this is eco R1. So I'll label it eco R1. So this one is eco R1. Eco RV and scal. I'll just call this one RV. It's eco RV and scal. And then this is eco R1 and eco RV. So let's say for this lane, we'll draw it out. So you're gonna have 2 fragments for this one, and you have the 2 cuts are these 2. So I'll draw the shape, circle. So you have 2 cuts. For instance, 1 cut right here and 1 cut right here. So this fragment is 2. And then this fragment is 8. And then it's the 2 cuts are made by the 2 restriction enzymes. Let's see. So the restriction, so this is lane 5, eco R1 and eco RB. So I'll call, what's it doing? So I'll call this one eco R1. R1, and this one is where eco RV cut. So you know that they're supposed to be RV. So you know that these are supposed to be 2 apart, 2. So then we'll look at the next lane. So we'll look at 6. So this is for 5. This is for lane 5. Now we'll look at lane 6. So I'll draw another circle. And these are a different combination of restriction enzymes. So then the restriction enzyme for that one, for column 6, for lane 6. So lane 6 is eco R1 and scal. So eco R1 and scal. So then for this, let's say like, oh, this looks like it's slightly bigger than 8, but it's still below 10. So we'll call this one 9. So this is 9. And then this one's really low, it's below 2, so we'll call it 1. So this adds up to 10. So then we could draw, so scal, which one was it again? It was, is lane 6, is eco R1 and scal. So eco R1 and scal. What am I doing? So eco R1 and scal. So eco R1 and scal. So I'll draw eco R1 right there. I'll draw. Eco R1 and then scal. So this is 1 and this big fragment on the outside is 9. So that's why you have those. So you know that Scout and EcoR1 are really close to each other, and they're way closer than EcoR1 and EcoRV. So we'll draw the third one to get a better idea. So now we just got to figure out, so we know where, by knowing where Scout is, and we want to draw Scout here, we got to know whether Scout is really close to this one. Well, it's 1 away from this one. We want to know it relative to this one. So EcoRV. So we want to know if it's on this side of it or this side of it. So we'll draw the third one, which is lane 7. And just based on lane 7, you know that these are pretty close in length. So we could say this one's like 3, and this one's probably 7. So that's for EcoRV and Scout. So they're pretty far away. So Scout and EcoRV are far away from each other. Further away than these. So you're gonna have, so I'll draw it. So Scout, and then EcoRV. RV. So then if you want to draw Scout here, you have to draw it far away from EcoRV. So this is probably 3, and this is 7. So then you know for sure that in this, if you want to draw Scout in this diagram, you can't draw it too close to EcoRV. So you have to be 3 away. So it's not going to be in between these, and it's going to have to be around 3 units. So it might be here. So that's 3 units away from here for Scout. And you have to check if it matches. So yeah, so it's 1, so it matches this one. So this would be one diagram you could draw. There's different ways of drawing it, as long as you know that Scout and EcoR1 are 1 unit away. EcoR1 and EcoRV are 2. This is 8, and Scout and EcoRV are 7 units away. And it makes sense because this part's 1, this is 2. This whole part all the way over here is 8, but this part from Scout, this is actually 7. So this right here.
I have a quick question, Carlos. So all we are doing is just drawing one diagram, or we are doing like multiple diagrams?
So the diagram that we want, I mean, you have room to draw multiple diagrams, but the diagram that we want is all 3 of them relative to each other. So we want to see how far is, where would all the cuts be if, like, let's say EcoR1. There's different ways of drawing it, but the only thing that's not going to change is these separations. So these separations are just estimates for the explanation. It's really messy, but this is all I have to explain. But yeah, so for sure, we want, this is going to be the same regardless, the distance from here is going to be the same regardless of where you draw the cuts. The distance between these two is going to be the same regardless of where you draw the cuts. Same thing for the distance between EcoRV and Scout. But drawing all 3 helps you get an idea of where to draw them relative to each other.
Okay.
So I'll upload this because this video was all over the place, but I'll upload this.
Yeah, sure.
And then so for this last ladder, so in order to confirm that we did this right, so then this one is around 7. This is 2, and this is 1. So yeah, that adds up 10. So you got your 7 fragment, your 2 fragment, and your 1 fragment. So this is referring, so these numbers are referring to the size. So that's pretty much saying if I labeled my ladder, like the 10 is right here, then 9 is right here, like right in between 10 and 8, and then 2 would be right here, and then 1 is down here. So that's if I label my ladder like that. You guys are going to label your ladders differently. Well, this is an example of how the analysis would, and this is a 3. It's a messy 3.
Basically, on the base of first letter, that's how we are determining the others, right? Like our 6, 7, 8, 5, 6, 7, 8 is determined on the base of first letter.
So 5, 6, 7, 8.
Yeah, because like what I'm saying is you have 10 right here, right? And all you did is you try to separate it out with the just revolving around that number 10, like 8 and 2, and then 2, 9 and 1.
Yes.
Basically, we are just looking for the first letter, and then that's how we determine the others.
Yes. So yeah, if once you determine these, you know that these have to add up to these because that's your full fragment. That's the whole fragment with one cut.
Are you talking about the number 1?
So this one is not as accurate. It's better to use 2, 3, 4 because they should match up perfectly because you're only cutting the plasmid in one spot. So if you cut the plasmid in one spot, you're just gonna get the same line. Regardless if you cut it here, or you cut it in any of these 3 spots, you're gonna get the same line, the same length. So yeah, use 2, 3, 4 to match up to get the number that these have to add up to. So these have to add up to these 3. So I'll use a different color to be a little bit more clear. So these 3 right here, these are the numbers you use to figure out. So these 2 have to add up to one of these. These have to add up to these numbers. Same thing with these and same thing with these. Since this is not a fragment, this is a plasmid, it's not cut up, you can't rely on this number. It's not correct. So yeah, you use these. And then depending on where you start off, like let's say, instead of starting off your diagram by drawing, like I drew E core 1 here, but I drew it here, but I could have drawn it here instead and then drawn this over there and it would still been right. But then it's just going to be relative to this. So if these could be switched as long as this is 1 unit away from here and this is 2 units away from here. It wouldn't be wrong as long as the distance between these 2 is always 2. Distance between these 2 is 1. Regardless of the, I guess the orientation you choose to use. I guess that's the confusing part because the orientation could be different. It's just the.
Yes.
I mean, you could use this orientation that I use I'm gonna upload this. So you could sort of use this orientation, but the sizes that you guys will have will be actually real sizes you guys determine from your ladder. So these are made up size. This is a made up ladder I made.
Because what I'm seeing, this 2, 3, and 4, their size gonna be same, right?
2, 3, 4, yes.
Right. So all we are doing is we are trying to get number pretty much close to these sizes for 5, 6, 7, and 8.
Yes.
Right. Okay,
Yes.
that makes sense. Got it.
I mean, they should add up. Like, so once you label your ladder, so you're gonna have your ladder labeled with all the numbers here. This one. It should match up. So this should match up to 10, and these numbers, these right here, should match up and add up to this number. So this number and this number should match up to the ladder, and then add up to this number right here, or one of these.
Okay.
Yeah. Yeah, I think class is over soon. You guys have any more questions? So I will upload this. I'll also upload the videos. And then the requirement for this lab is a full lab report. So with introduction, results, conclusion, all that stuff. It'll be, you'll need to include that on the results. And also, what's it called? I'm trying to think. What else was I gonna tell you guys? So it's a full lab report. Oh yeah, office hours. So I made an announcement just in case you haven't read it. Office hours are still Tuesdays and Wednesdays from 2 to 3. It'll be through a Zoom link, which I included in one of the announcements I made. So in case you guys have more questions or more clarification on like the analysis portion, you guys could just click on the Zoom link from 2 to 3 on Tuesday, Wednesday. And there shouldn't be any password on there. So you guys could just join just by clicking on the link during those times. And I mean, you guys have any questions, feel free, but I think I'll probably end it here. So stop sharing screen. I don't know how to stop recording, but yeah, I think that's it for this class. So I'll upload the video. I'll send the guys an announcement. I'll also give you guys the links to the videos that I included on here also. So I think, yeah, that's it. So class is done. Yeah, so you guys are dismissed. And I don't know how to stop the recording.